Hello, my name is Laura Tung. I'm going to present our paper co-authored with Dr. Carl Kingsford, Practical Selection of Representative Sets of RNA-seq Samples Using a Hierarchical Approach. A vast number of RNA-seq samples are available at large sequence databases such as the SRA. However, most RNA-seq analysis tools are evaluated on limited RNA-seq samples, so the tools may not be sufficiently evaluated with a variety of cell tissue types and disease conditions. This drives the need to select a representative subset of RNA-seq samples that effectively summarizes a large collection of RNA-seq samples to capture various essential transcriptional phenomena. The selected representative subset not only can facilitate comprehensive, unbiased evaluation of bioinformatics tools, but also can benefit the parameter optimization in bioinformatics tools. So our objective is to develop a computational method of selecting a representative subset from a large collection of RNA-seq samples, such as the SRA. Here, we symbolize each RNA-seq sample as a data point, and the distances between them signify how dissimilar these RNA-seq samples are. Samples with rare cell tissue types or disease conditions are far away from other samples. Basically, representative set selection methods find a subset of data points to efficiently describe the original data collection. An easy way to select a subset is random sampling. However, a randomly sampled subset tends to follow the density distribution of the original set, so samples of rare cell tissue types are not sufficiently represented. In contrast, a representative set captures significant information in the original set more efficiently, and it has low redundancy among representatives. Each sample in the full set is represented by its closest representative, and these lonely samples over here are selected as representatives themselves, so samples of rare cell tissue types are sufficiently represented. In the field of RNA-seq analysis, a geometric sketching algorithm has been developed for single-cell RNA-seq to select a representative subset of cells. And a Python package, Apricot, was recently developed for selecting representative sets using submodular optimization. Both geometric sketching and Apricot select a representative set that approximately evenly spans the space of the original data. For RNA-seq analyses, even coverage of the transcriptional space is important in order to represent rare cell tissue types, as we can see here. While geometric sketching requires knowing samples' gene expression vectors, Apricot can work with the similarity matrix between samples directly. In our representative set selection problem, given a large set of RNA-seq samples for a given organism, we want to find a representative subset of RNA-seq samples that minimizes the maximum distance between each sample and its closest representative sample. Here, the red line is the maximum distance. We want to minimize it. To perform representative set selection, one needs to obtain gene expression vectors of, or distances between, all RNA-seq samples in the SRA. However, it is infeasible to download all sequences of all RNA-seq samples from the SRA to obtain their gene expressions. Thus, we use a sequence-based approach for representative set selection by randomly sampling a small subset of reads from each sample. Since the frequency of K-Mers can be used as a signature of the underlying sequence, the similarity between K-Mer distributions of RNA-seq samples reflects their sequence similarity. Thus, K-Mer similarity is a reasonable approach for measuring the distance between samples. So by counting the K-Mers of RNA-seq reads using jellyfish, we select a representative set based on the K-Mer similarities between samples. We use cosine similarity as the K-Mer similarity measure, as K-Mer vectors have similar aspects to TF-IDF vectors, and cosine similarity is a good measure for K-Mer-based metagenome comparisons. Choosing a proper K-Mer size is important, as smaller K-Mers give less info about sequence similarities, while larger K-Mers may result in fewer matches due to sequencing errors. Thus, by plotting the number of distinct K-Mers versus K-Mer size for different read lengths, we choose an optimal K-Mer size 17, which is a compromise among the optimal K-Mer sizes for different read lengths. Because of the large number of RNA-seq samples in the SRA and the huge number of K-Mers in each sample, there are memory and runtime challenges in computing the full similarity matrix. To tackle this challenge, we developed a novel method called hierarchical representative set selection. It is a divide and conquer-like algorithm that breaks the representative set selection into subselections and selects representative samples through multiple levels. In the hierarchical representative set selection algorithm, at each level, the full set is divided into equal-sized chunks using a seeded chunking method, which I'm going to describe later. The similarity matrix for each chunk is computed, and weights that determine the size of the representative set for each chunk are computed. Representative set selection is then performed on each chunk, for example, using Apricot's facility location approach. The representative samples selected from every chunk are merged into a new set, which becomes the full set of the next level. This process iterates until the size of the similarity matrix of the merged set is feasible for the computing resources. Then the similarity matrix of the final merged set is computed, and representative set selection is performed on it to get the final representative set of a desired size. Here is an illustration of applying hierarchical representative set selection to the entire set of human RNA-seq samples in the SRA, which has almost 200,000 samples. We use two levels of divide and merge. The first level has 197 chunks, and the second level has 40 chunks. Each chunk at both levels has 1,000 samples. About 200 representative samples are selected from each chunk. The final merge set has nearly 8,000 samples, which is feasible for our computing resources to compute the similarity matrix. And smaller size representative set selection can be performed on this similarity matrix. When chunks have no overlapping regions, the union of the representative sets selected from every chunk would be similar to the representative set selected directly from the full set. This can be seen from this two-chunk example. Here, the red circled dots are the selected representative data points. When chunks have overlapping regions, unnecessarily more representative data points may be selected in the overlapping regions, which may cause other regions to have less quota for representative data points, and thus to be less represented. The chunk overlap means data points and their close neighbors are in different chunks. For example, different chunks occupy the same cluster, as shown in this two-chunk case. We can see that the resulting representative set is less optimal in this case. Thus, more separated chunks lead to more accurate hierarchical selection. So the hierarchical selection method leads to a new challenge. How do we divide samples into well-separated chunks without knowing all the distances between all the samples? To solve this challenge, we developed a seeded chunking method that can generate well-separated chunks. The seeded chunking method first finds far-away seeds for chunks. These two circle dots are the two seeds for the two chunks in this example, and it lets closer samples go into the same chunk if possible. The seeded chunking method first chooses a seed for each chunk such that the seeds are farthest away from each other. To avoid extra computational cost in the seed selection process due to the full set not fitting into memory at once, we randomly select a subset from the full set and choose seeds from this subset. The seeded chunking method uses the farthest point sampling to find the seeds for all chunks. That is, starting from a randomly selected seed, the seeds are chosen one at a time, such that each new seed has the largest distance to the set of already selected seeds. This farthest point sampling is performed on the aforementioned randomly selected subset. Then each sample in the full set is assigned to its closest seed. Since chunks have equal sizes, the sample is assigned to its closest seed among all currently non-full chunks. Now that we solved the challenge of chunking by using a seeded chunking method, we have another challenge. How do we determine how many representative samples to select from each chunk? Denser chunks, in which samples are more similar to each other, should have fewer representative samples than sparser chunks, since we want representative data points to approximately evenly span the space of the original data, so that rare cell tissue types can be sufficiently represented. Since chunks have equal sizes, denser chunks occupy smaller spaces than sparser chunks. Thus, we designed a mean squared weighting scheme to assign the representative set size to each chunk based on their average density or sparseness. In the mean squared weighting scheme, the representative set size for each chunk is proportional to the square of the mean distance between the samples in the chunk. When the full set size divided by the chunk size is not an integer, one or two chunks are not full. Thus, the representative set size for each chunk is also proportional to the ratio of its actual chunk size over the full chunk size. Hausdorff distance can be used to evaluate how well a selected subset represents the full set. A smaller value is better. However, classical Hausdorff is highly sensitive to extreme outliers. In this example, an extreme outlier makes the classical Hausdorff very large, although all data points except for the outlier are well represented. Thus, in practice, partial Hausdorff distance is used to evaluate the representative set, using the kth largest distance to replace the maximum distance in classical Hausdorff. Partial Hausdorff distance has a parameter q. When q is small enough, partial Hausdorff is very close to classical Hausdorff, but is robust to extreme outliers. In this example, the second largest distance is used, so the extreme outlier does not affect the partial Hausdorff. We use partial Hausdorff DHK to evaluate the summarization quality of representative set selection. A smaller value indicates the representative set is better. We refer to direct representative set selection using apricot as direct apricot, which includes 1. Computing the full similarity matrix, and 2. Applying apricot. The computational cost of direct apricot mainly comes from part 1. In this implementation of hierarchical selection, we use apricot as the base level for representative set selection on each chunk and the merged set. We compared the performance between direct apricot, hierarchical selection, and random sampling using the most recent early time and mid-time samples in the SRA as different sizes of full sets, up to 12,000 samples. The early and mid-time samples are the ones in the earliest quarter and the middle of the SRA time span. Hierarchical selection achieves summarization quality close to direct apricot. Here is the DHK result of using the most recent samples. A low DHK value is desirable. Hierarchical selection initially performs better than direct apricot and then is modestly less accurate than direct apricot for larger full sets, but their DHK is fairly close. Our results of using early time and mid-time samples in the SRA are not shown here, but the conclusion is the same. Overall, the average percent increase in DHK of hierarchical selection versus direct apricot is 5.37%. And importantly, the summarization quality difference between hierarchical selection and direct apricot does not get larger when the full set size further increases. Hierarchical selection substantially reduces the runtime and memory usage of direct apricot. Here is the runtime and memory reduction with hierarchical selection over direct apricot using the most recent samples. We also have the results for using early time and mid-time samples, which are not shown here. As the full set size increases, the runtime reduction and memory reduction generally increase. The runtime reduction can reach 8.4 times for 10,000 samples, and the memory reduction can reach 5.35 times for 12,000 samples. For the entire SRA set of human RNA-seq samples, so 200,000 samples, the estimated runtime and memory usage of direct apricot are 66 days and 2.3 terabytes, as we extrapolated based on its time and space complexities. This is infeasible. Our hierarchical selection on the entire SRA set takes only 17.6 hours and 56.4 gigs of memory to complete this task, making this task fully feasible. We use 85 cores for all cases, so the estimated runtime reduction with hierarchical selection is 90 times and memory reduction is 41 times. Hierarchical selection outperforms random sampling for the entire SRA RNA-seq set. When selecting 7,000 representative samples, hierarchical selection outperforms random selection substantially. Random sampling's DHK values are almost 1.0, that is the maximum cosine distance, and do not change, indicating its poor performance on the entire SRA set. These results show that hierarchical selection offers a practical solution to representative set selection on large databases like the SRA. High quality and efficient representative set selection is possible, and we showed that through hierarchical selection. Our novel hierarchical representative set selection method can greatly reduce the runtime and memory of direct representative set selection with full similarity matrix computation, while the hierarchical selection achieves summarization quality close to direct representative set selection. Hierarchical selection substantially outperforms random sampling for the entire set of SRA human RNA-seq samples. This is the first approach to this problem that can scale to collections of the size of the full set of human RNA-seq samples in the SRA. We would like to thank Dr. Guillaume Marseille for helping improve the speed of per-pair computation of K-Mer similarities significantly. We also thank the Kingsford Group members. This work was supported by the NSF Graduate Research Fellowship Program, the US NSF, the Gordon and Betty Moore Foundation, and the US NIH. Thank you for listening.